afternoon, everyone. So today I'm going to share with you some of the things I think it's relevant because this is a really nice campus. Every time I think about your campus, I think about it's really hard to reach. Really hard to reach. Like if I'm on campus, I have to have a car or I have to have the a bike or a motorcycle to commute between buildings. But this is really nice, and I think it's connected to wildlife conservation and everything. So I want to begin with how I want to introduce these topics because I have a friend introduce this bird to me. So this is passenger pigeon. So I have a friend in National Taiwan University. So one day he talked with me about this passenger pigeon. And the story he told me is probably some of you already learned. This is the last passenger pigeon in the world. And it's died in the zoo very sadly. And there's nothing. So he said, we have extinct the animal all the time. But this is in our history, so we know exactly. But more astonishing is they used to be a lot. So used to has tons of millions of the birds in North America. But it went extinct suddenly during human history. So this is how I heard the story from my friend. And actually, when he told me the story, it's the time he started design the experiment to understand how a species went extinct in the human history or in the past, because he can get samples from the museum. So they decide to use the museum sample to assess the genomic diversity. And of course, a few years later, and this is a paper they published. So this is a paper they published in PNAS and talk about what's the genetic diversity exists in their genome data and how they try to understand the population size in the past for this species. And just to show you one of their figures, they try to use the genetic diversity data, try to see what's the effective population size in the past. So in the x-axis are the time, and the y-axis is the effective population size, like what's the number of birds contribute to the next generation genetic material. So this is what they use their data to predict. So this is how I get interest. Not only because we can count number of individuals in the field, but also use genetic data, try to understand the history of either animal or plants. So why this is important? This is important is because we have many species, but some species are really abundant. So usually when the species are really abundant around us, we don't pay attention. I think they are just of course they come every day. But even species with huge number could be an endangered species in a few years, which was not expected. So usually when we think about what other species are endangered, other species with very few numbers, right? We'll see some of the examples later. And to understand the historical reason the species went extinct might help us to understand what can we do better for conservation. So this is how I get involved in this field and also try dig out the literature, try to understand how people study that. So I'm to ask a question here. Do anyone know how many giraffe species are there, on Earth? So how many species do we have? 70? Okay, good. Any other number? 90? Okay. 14. Laura? 8. Lower? 2. Higher. Okay, good. So you see, I think I like Professor Song's approach. We get answer right away. Usually you have to wait and tell students, this is a question, I'm waiting for your answer. I don't have to do this today. So the approach they took is they try to get the population. So this is a distribution of different populations. So we used to name the giraffe with many different species and subspecies. And of course, for conservation reason, we always count the number of individuals or estimate the number of individuals for each population. So here is the list. So you can see this is the table I get from the paper. They have the many species and subspecies this here and has their common name next to it. And you have the population and distribution. And so this is correspond to the previous figure. Then this is the population size. So this population size is the estimate from the wildlife study. And when we think about whether a species is endangered, then we say, okay, let's see how many individuals can we see in the field, how many individuals we can estimate from our scientific data. If the number gets too low, they say, oh, this is important, so we have to list all the species and we have to put conservation effort on it. So this is how we do conservation most of the time. So what this group did was getting samples from this population and try to understand whether those individuals share the same or similar genetic materials. And using the genetic data, try to see whether they can form different groups. So this is like a traditional taxonomist. They measure everything, then try to see whether the individual or a group of individuals are similar or dissimilar. So this is what they show in the paper. So for the colors here are the individual samples corresponding to the location in the map. But I want you to pay attention to the clusters. So for the clusters are the samples that close with each other. So it means those individuals, for example here, they are very similar to each other. They form a cluster. So if that's the case, then we say, okay, all these individuals or individuals from the populations, they share similar genetic materials. We can cluster in or identify them, group them to a single species. So this is how 2 years ago, so this is a paper in the beginning of 2016, then they say the headline news for this research is there are only 4 giraffe species. Then not all scientists agree with this. So some scientists challenge what do we think about different species. But this is a really nice study I like to share with everyone here, and also sometimes I talked to my students as well. Because in the previous passenger pigeon, they do the whole genome sequence. So because the technology advanced, people think about genome sequence become cheaper and cheaper, but still very expensive. You think about to conduct the entire genome. And the nice thing about this study is they use countable molecular markers. They didn't really study the whole genome. They might do that later, but for this particular paper, they only use 7 nuclear markers. Try to understand the genetic relationship among all the individuals they sample. And why this is important, or I should show you the next slide. So this is like using different methods. So if you use genetic data to classify, and there are different methodologies you can use. So use the different methods, they can get like very similar results. The authors also show another analysis. So this analysis, you can see very similar to the previous one, and this is called structural analysis. So given the genetic information from our sample, can we try to understand what's the best estimate about all these individuals? So it means like how we can group them together. So based on this, you can see there are 4 different clusters in different color. So it means all the individuals in our sample ensures similarity in 4 different groups. So this is the basic, and they use another analysis called PCA analysis, principal component analysis, to show very similar results. So this paper used a small number of markers and get huge number of individuals. Then they try to understand the number of species, or at least number of distinct genetic groups in the giraffe population in Africa. Why this is important? Because we used to think each population or each subspecies are quite unique. And if that's the case, then the subspecies level, they are all in different populations. But we think we group them into a single species or just a few species. But this study tells us this is probably something we have to rethink about it. So in the past, we think they are probably all giraffe. But maybe, some of the subspecies are more close to each other than the other groups. So in this paper, although still, debating, but the authors suggest we probably can separate all the giraffe species into 4 different species. Then they also estimate the divergence time. So this is also used genetic data to see how different some of the species. So do pairwise comparison and showing, so these 2 are the most closely related group. And like some time ago, they split into 2. So they share the common ancestor, then they split into 2 different species. Okay, so this is about the species. And why this is important? So the same thing, why scientists want to study, why we should care whether it's a single species or 2 species or 10 species. So the important thing is, when we talk about conservation, of course, like deep in the rule, conservation is important because we want to protect the environment, the entire ecosystem. But it's very hard because the entire ecosystem is huge and we don't even know how many species are in there. To emphasize the importance of the conservation, we have to have identify some species, right? So here is like they think all the giraffe species in Africa, belong to 4 different groups. Then we should go back to the initial counts, right? Count how many individuals are in each group. Are some of those unique genetic groups are endangered? Means the number is very low, and we should really emphasize on that. Are some of those are the single species and distribute in different areas, but their genetic material are very similar. So this is how today we can use some genetic information to help us to to study or to help us the wildlife conservation. Okay, so the second question is, do anyone know how many elephant species in the wild? I think this is much simpler. How many elephant species do we know? 20? 1? 10? Remember your textbook? I thought my textbook always tell you. Okay, so when I was in, I think, undergraduate. So when I was an undergraduate student, so I know there's elephant, Asian elephant, and African elephant, right? Yeah, people not good. And also my teacher said, if you look at the ear of the elephant, and you know the name of the elephant. So that's how I learned it. But scientists are like debating because they do a lot of studies, not only molecular, even before we have all the molecular data. So they debate how many species exist in Africa. So are there two distinct species or just one? I study speciation questions, so I'm very interested in how people define species because it's a very difficult question. In my research, I use Drosophila to do this research because it's easy to study in a laboratory. In the field, of course, I want to have adventure in Africa, but it's usually very, very hard to do all the field work. So when scientists start wondering whether the different group of elephants are different species, so they try to do a lot of things, do measurement, do like life history studies, and they also show the different groups have different reproductive time. So all this information leads the scientists to rethink about there's only one African elephant. So actually they probably two different species in Africa. And earlier this year, there's another study. So the importance of this study is they try to understand the entire history of elephants, not only from the current individuals we can access, right? For the giraffe study, we just get the sample from the field, we collect the sample from the field and try to understand the genetic relationship. In this study, they use some modern technology to study the ancient DNA, just like my friend did it with passenger pigeon. So they dig out not only the elephant species or elephant populations in natural, but they also put the specimens. Just to put the story short, you can see in this figure, so this is a figure just published this year. So they put not only the species here, so here you can see the Asian elephant and also two species of African elephants. They also have mammoths and some other extinct species in this tree. So this is a typical phylogenetic tree. Try to understand the relationship among different species. And this paper use all this information, try to dig out or try to understand how many species are in Africa and what's the relationship. They use the genetic information and try to understand how long ago these two elephant species separate with each other. So if I remember correctly, it's 500,000 years ago. These two already separate. They are genetically isolated from each other for a very, very long time. And this is very strong evidence in addition to all the characteristic measurement, all the life history measurement, and this genetic data also in agreement with all the previous results. We think the elephant, I think most of people agree the African elephants can be separated into two separate species. So for all these studies, actually there are essential concepts within that. So I teach undergraduate biology genetics. So in the undergraduate biology course, we usually discuss what is a species. So how we define species. It's not that easy. Biologists have different ways to characterize species. Most of the time, no matter which type of data you used, I think the data always agree with each other. Because like I have many friends, I graduated from Department of Entomology. I have many friends, they are working as a taxonomist. They describe species using morphology data. And the morphology, appearance of an individual was a lot of was controlled in the genome. So even when we using the genetic data, most of them, they agree with each other. The data are in agreement with each other. And the other thing practically we do is phylogenetic reconstruction, use characters and try to understand how those characters can separate the population of the individuals into different groups, or we can cluster them into a single group. Then there's a whole bunch of different methodologies under this category, and some people or a lot of scientists agree to use monophyletic group as a separate species. So they will use define the monophyletic group as a single species. And when we have a lot of genetic data, so usually people will think about whether we are using the genetic data to construct species tree. That's what we're thinking about. We're trying to understand the phylogenetic relationship among different species. But actually the data we use are from the genome. So actually we are reconstructing gene trees each time. So especially when you use just a specific marker, and our tree, our phylogenetic analysis reflects the history of the gene instead of the history of the species. If we use enough molecular marker, and we probably can take the majority to reflect the species tree, as a lot of people now use whole genome data or whole genome random sample SNPs, try to understand the species phylogeny. And the third level, which we didn't really talk a lot, actually when I show the figures and all the data on the table, actually there's quite a lot of calculations. For example, I put some of the terms here because people try to understand the genetic diversity. So within a population, how individuals differ from each other. Like within this classroom, we have quite a diversity in morphology, in our appearance, but deep in our genome, the genetic diversity in the whole human population are very, very small. Then we also mentioned about effective population size. So I start from the passenger pigeon, talk about effective population size fluctuate a lot for this particular species in the past. And human population will have a huge expansion. Then this is how we mention in population genetics, we like to talk about how many individuals contribute to the gene pool in the coming generations. And that's what we mean by effective population size. Then the other thing we didn't talk a lot is population admixture. So in human is a great example. So if you, from the typical textbook, human population originates in Africa, we migrate out of Africa to the Eurasia and also Americas. But there's also population admixture, especially in Northern Africa. So people will come back to North Africa, they admix there. When we say population admixture means the population has diverged a little bit, but they come back, interbreed again. There's a genetic information exchange in the population, and we can detect that as well. So given these examples and the concept here, I want to sort of emphasize with the genome data, with the molecular tools, what we can do more about sort of the ecological or wildlife study. So I want to have this huge table. So this is just part of the table, but I want to go through this table gradually. Okay, so this is a huge table. This is on the top. When we want to study, we think about what kind of data do we have. Do we have the whole genome data, or we just have a subset of the genetic information? Then we can go through the questions here and all the calculations. Like I talked about genetic diversity here, right? So in the previous slide. And we think about whether we want to understand the population structure. Then you can go through the path to know what type of analysis you would like to do and what you can obtain from the genetic information from the data. And sometimes the data can be combined with, for example, the morphological information or the ecological information. And at the end, we'll be able to understand some of the important genetic information's control traits, like the morphology or life history. So this complicated slides, I think you can go back home and to think about it. And in each step, they guide this slide guide us to like what should we learn or what should we obtain or what type of analysis we should do. And actually, it's a very good, I think, the tool like guide to sort of like for this type of study, so molecular ecology or conservation biology. And this is the end of the slide. Try to sort of, okay, at the end, we want to associate our study to environmental factors or the environmental like influence, and we try to understand how those two interact with each other. To end my talk here is try to think about another thing. So I start with the passenger pigeon, and it's extinct. Then we have many species extinct, and the number of species is sort of like undergo that path. So what can we do? Some very smart and intelligent scientists would say, can we get them back? So there's a term called de-extinction. And if you type de-extinction words, and you will see many discussions. And this is just for us to think. I don't have an answer, because if you ask me, they will say, I don't think that's a good idea. Yeah, but people are doing it. People are trying to clone mammoths and some other species, and some experiments has been done. Then we just have to wait to see. But it's just not a very good idea. Instead of trying to bring all these extinct species back on Earth, I think using part of the money, we can do better for environmental issues, for conservation biology. So this is just to end today's talk and to allow you to think about how we will do with all the environmental issues and all the sort of the conservation and wildlife biology questions. And thank you.